In this video, we will look at the components and setup for translation. The base definition of translation is interpreting one language into another, and this is essentially what we are doing in this process in our cells. We are translating the genetic code of nucleic acids into the protein codes of amino acids. Genetic code is translated in three base pair chunks called codons. This process is complex and involves many components, including at least three types of RNA. Let's explore the structure and roles of the first two here, tRNA and rRNA. tRNA plays a direct role in the process of translation. Correctly hypothesized by Francis Crick of double helix fame, in the 1950s, tRNA has two main roles in translation, which are recognizing the three base codon in mRNA and carrying a specific amino acid to that codon. tRNAs recognize codons via complementary binding of a region of them called the anticodon. This anticodon follows all the same rules of base pairing we have previously seen, including binding in an anti-parallel direction. Different tRNAs carry different amino acids, and they are thus named for the amino acid that they carry. If we zoom in on the structure of tRNAs a bit more, we see they have a distinctive cloverleaf pattern containing three stem loops, some variable sites, a 3' end where the sequence ACC has been ligated on, which will serve as our acceptor site for the amino acid. And along with all of this, our tRNA contains several modified bases. tRNAs serve the important role of connecting the appropriate amino acid with the appropriate codon, but they don't come preloaded with the correct amino acid. That important job falls to an enzyme called amino acyl tRNA synthetase, of which there are 20 of them. The basics of this process are that in a two-step process, this enzyme matches an amino acid with the appropriate tRNA while expending a bit of energy. This process is remarkably precise and only pairs up the wrong tRNA and amino acid about 1 in 10,000 times. Once the tRNA is charged, it can proceed to finding the appropriate codon with which to match. At this point, if you are a keen observer, you may have noted something. Only 20 tRNAs, and thus 20 amino acids, does not cover the number of combinations possible for our three base pair codons, which is actually 64 combinations. However, this brings us to an important quality of our genetic code. It is degenerate. This means that sometimes there is flexibility in the match between codon and anticodon base pairs. This usually occurs in the third codon position, though there are three exceptions to that. Francis Crick was again the one to propose this quality and termed it the wobble rule. So, in many cases, the identity of the third base in the codon will not affect the amino acid that is matched. This has all sorts of implications, which we will not go into here, but suffice to say, the wobble rule is an important quality of our genetic code. Our second important RNA structure in translation is the ribosome. This is a complex of rRNA and proteins that is typically divided into two subunits, a large and a small subunit. These subunits are typically assigned numbers based on their size, but we won't get into that for now. In eukaryotes, when we refer to ribosomes, we typically mean the ones in the cytoplasm or attached to the endoplasmic reticulum, even though we actually have a second type in our mitochondria and in chloroplasts if you're a plant. For the purposes of translation, we will highlight three important sites: the peptidyl site, or P site, the amino acyl site, or A site, and the exit site, or E site. In the next video, we will see what happens at each of these sites as we progress through the process of translation.